Welcome students. So, in this period we are going to study about the process of oogenesis. So, we have already studied the spermatogenesis. See here, what is oogenesis? It is the process of formation of haploid female gamete, that is egg or ovum, from the diploid germinal epithelium. Okay. So, from the diploid germinal epithelium, actually you know that the ovary is lined by germinal epithelial cells. Now, these epithelial cells give rise to haploid female gamete. This process is called as oogenesis. Now, see here, like that of the spermatogenesis, this oogenesis is also divided into 3 stages. Same names are there, that is the first phase is multiplication phase, second phase is growth phase and third phase is maturation phase. Same phases are there. So, see here, I will show you the diagrams actually from the textbook. Okay. See here students, this is the process of oogenesis. Now, see here, 3 steps are there. Okay. Now, here the first step, that is multiplication phase. See here student, this is the short form actually, these are the primary germinal cells. PGC means primary germinal cells, which are diploid in nature. They undergo multiplication phase, means they show in this phase repeated mitotic divisions. So, one cell divide mitotically into 2 cells and these 2 cells again divide mitotically each into 2. So, from one primary germinal cell, see here, develops 4 new cells called as oogonia. Okay. So, from one primary germinal cell, by repeated mitotic division, there is formation of 4 oogonia. Okay. Now, out of this oogonia, some oogonia they enter in growth phase. These oogonial cells actually they stop dividing and they grow in size, they increase in size, and here that cell is now called as primary oocyte. Okay. No doubt primary oocyte, like that of the oogonial cell, it is diploid in nature. So, 2n having 2 sets of chromosomes, that is 46 chromosomes, they are present in the primary oocyte. Now, this phase is called as growth phase, that is the growth takes place. कशाची ग्रोथ होते? ओगोनियल सेलची ग्रोथ होते. कशामध्ये? प्रायमरी oocyteमध्ये, म्हणून याला म्हटलेले ग्रोथ फेज. ओके. नाउ द थर्ड फेज इज मॅच्युरेशन फेज. See here, what happens? This primary oocyte undergo meiosis type of cell division. So, you know that meiosis, it is completed in 2 steps, meiosis 1 and meiosis 2. So, first of all, primary oocyte which is diploid, it undergoes meiosis 1 and it divides into 2 cells. See here, you can observe in the diagram, there is unequal division of the primary oocyte. Unequal म्हणजे याच्यावर 2 सारख्या सेल्स तयार होत नाहीत. बरोबर आहे. या ठिकाणी एक सेल आहे लार्ज सेल आणि दुसरी आहे स्मॉल सेल. सो द लार्ज सेल इज कॉल्ड एज सेकंडरी oocyte. म्हणजे प्रायमरी oocyte पासून सेकंडरी oocyte तयार झाला and the small cell is called as first polar body. Okay, this is the small cell called as first polar body. Now see here, what happens? This secondary oocyte, it enters now in meiosis 2. Here meiosis 1 is completed. Now the secondary oocyte, it enters in the meiosis 2. But see here, the meiosis 2 is not completed. Here the secondary oocyte, though it starts meiosis 2, here the division takes place only up to metaphase 2. Okay, only metaphase 2, this phase is observed in the secondary oocyte and here it is stopped. Okay, the further divisions are stopped in the oocyte or further divisions are arrested. Okay. Now such type of secondary oocyte, it is released, it is ovulated from the Graafian follicle. So if you remember, we have studied the menstrual cycle. At that time I told you that on 14th day of the menstrual cycle, the secondary oocyte, it is released. Graafian follicle breaks down and releases the secondary oocyte in the peritoneal cavity. So this secondary oocyte, now in which the cell meiosis II is arrested at the metaphase II. This secondary oocyte is liberated. Okay. Now if you remember the female reproductive system, okay, I will show you students. See here, okay, we have studied this. Now see here, this is the Graafian follicle. Now this Graafian follicle releases the secondary oocyte. Now you know that this secondary oocyte, by this fimbriae, which are finger like structure, this is directed, it is drive towards the infundibulum, that is oviduct. Now you know that this part is called as oviduct or fallopian tube. Now this secondary oocyte, which is liberated on 14th day, now it is directed towards the oviduct and it is caught by the fimbriae. So fimbriae catch that secondary oocyte and drive it towards the infundibulum. Now you know that here there is opening called as ostium. Now this oocyte enters through the infundibulum and here it comes into the ampulla. Okay. So this is the secondary oocyte. Okay. Now in this secondary oocyte, the meiosis II, okay, it is only up to the metaphase II. Here now the ampulla, and I have also told you that ampulla is the site of fertilization. इथपर्यंत स्ट्रक्चर लक्षात आलं. आता हे एम्प्युला मध्ये आल्यावर काय होतं, इथपर्यंत आलेलं आहे आपलं सेकंडरी oocyte. आता समजा आता या ठिकाणी बघा, now see here the secondary oocyte which is ovulated now, this secondary oocyte, okay. Now see here, this secondary oocyte enters in the ampulla. आता मी तुम्हाला आकृती दाखवली ना, तर एम्प्युला मध्ये गेल्यानंतर राहिलेल्या ज्या स्टेजेस आहेत, म्हणजे मेटाफेज नंतर काय असतं, मेटाफेज नंतर ॲनाफेज, आता मेटाफेज 2, नंतर ॲनाफेज 2, नंतर टिलोफेज 2 आणि नंतर सायटोकायनेसिस 2. तर ह्या ज्या राहिलेल्या अवस्था आहेत की नाही, ह्या एम्प्युलामध्ये पूर्ण होतात. पण केव्हा, व्हेन द स्पर्म एंटर्स इन द सेकंडरी oocyte. Okay, when the process of fertilization starts, when the sperm enters in the secondary oocyte, what happens at that time in the ampulla, the remaining meiosis II, it is completed, and then the secondary oocyte, it divides into two at the end of meiosis II. It divides into two. Again see here, again there is unequal division. Secondary oocyte divides into two. One is larger cell called as ovum. आता हा egg आहे किंवा हा ovum आहे, and the smaller, now this small cell is called as second polar body. आपण इथं बघितलं, प्रायमरी oocyte पासून फर्स्ट पोलार बॉडी बाहेर टाकली जाते, सेकंडरी oocyte मधून सेकंड पोलार बॉडी बाहेर टाकली जाते. आता हा जो फर्स्ट पोलार बॉडी ना, तो सुद्धा meiosis II दाखवतो आणि त्याच्यापासून सुद्धा दोन पोलार बॉडी तयार होतात. म्हणजे समजा या ठिकाणी आपण म्हणू शकतो की 3 पोलार बॉडी या ठिकाणी आपण बघू शकतो. बट नॉर्मली फर्स्ट पोलार बॉडी डू नॉट एंटर इन द meiosis II. इथे जरी दाखवलं आहे तर जे नॉर्मली इट डू नॉट एंटर इन टू द meiosis II. इन दिस वे द सेकंडरी oocyte gives rise to ovum, and here the smaller one is called as polar body. Okay student, so this is about the प्रोसेस ऑफ oogenesis. आता तुम्हाला फरक लक्षात आला असेल, स्पर्मॅटोगोनिया आणि oogonia याच्यामध्ये काय फरक आहे बघा. इथं आपण बघा, एका oogonia आपल्याला एकच एग मिळाला की नाही, एक oogonia डेव्हलप झाला अशा प्रकारे आणि त्याच्यापासून आपल्याला एकच एग मिळाला आणि इथं अनइक्वल डिव्हिजन झाली प्रत्येक वेळेस आहे ना. आता थोडंसं इकडे बघा बरं, इथे काय दिसते आपल्याला, इथे काय झालेलं आहे, आता हा प्रायमरी स्पर्मॅटोसाइट आहे, त्याचे इक्वल डिव्हिजन झालेले आहे की नाही. दोन्ही सेकंडरी स्पर्मॅटोसाइट सेम आहेत, पण oogenesisमध्ये आपण बघितलं एक सेल मोठी तयार झाली आणि एक सेल छोटी तयार झाली आणि छोट्याला आपण पोलर बॉडी म्हटलं, हो की नाही. आणि इथे सेकंडरी स्पर्मॅटोसाइट जे आहे ते सुद्धा इक्वल डिव्हिजननी स्पर्मेटिड्स तयार करतात. तिकडे आपण बघितलं की इथे एक आधी मोठी सेल होते आणि एक छोटी सेल होते आणि म्हणून हा एक फरक बघू शकता तुम्ही की इथं आहे की नाही एका स्पर्मॅटोगोनियापासून किती स्पर्म मिळाले आपल्याला, चार स्पर्म मिळाले है ना. परंतु इथे या ठिकाणी बघा एका उगोनियल सेलपासून आहे ना, एका उगोनियल सेलपासून आपल्याला एकच एक मिळाला, एकच ओहम मिळाला. हा फरक तुम्ही लक्षात ठेवा. So okay student, so all this is about the process of oogenesis. Okay student, now in the next period we will see about the structure of the ovum.